So thank you, Tamar, and thank you to the organisers for the invitation to come and speak to you today about method validation studies, and in particular about planning studies. So in the first session, Lawrence Seberson gave a very interesting overview about the validation process and some of the issues relating to accreditation. And in this session, I'm going to very much focus on the planning side of things and the importance of planning, what might go into a method validation plan, and some of the experimental designs that we might use. And then I'll just touch on some of the Eurachem guidance that's available. So why is it so important to plan our validation studies? Well, it's potentially quite a complex activity and it can generate quite a significant amount of data. So really, we want to try and do things as efficiently as we can. And there are many decisions that we have to make when we're thinking about a validation study. So things like which performance characteristics are going to be important, which materials we might analyse, how many replicates are needed, and what we're going to actually do with the data once we've got it. And of course, very importantly, how are we actually going to determine whether our particular method is actually fit for purpose? So to ensure the validation study itself is fit for purpose, we really need to address all of these issues before we actually start work in the laboratory. And the advantage of using a validation planning template, which I'm going to talk about briefly, is that it allows a very consistent approach. And also at the end of the process, that plan could be fairly easily converted into a validation report. So what might go into a validation plan? Well, this is just some guidelines about what you might cover. There's obviously different ways you could do that, and I'm sure some of you might already have templates available in your laboratories, but I think this is the minimum of the issues that you should address. So we need to be clear about which method we're validating, the status of that method, and why we're actually carrying out this particular validation study. We need to think about the analytical requirement. So why are the measurements being made? What will our data be used for? We need to consider the performance characteristics we're going to study, and ideally have some idea of some performance targets. So if we decide that, for example, precision is important, once we've evaluated our precision, how are we going to decide whether that precision is acceptable? So ideally, we need something to judge the method performance against. It's useful then to include some kind of summary of the outcome of the validation study, and then there should be a final approval. So really the validation isn't complete until somebody has taken a decision as to whether or not the method is actually fit for its intended use. So I'm just going to look briefly at each of these points in turn. So we do obviously need to be clear what it is we're actually validating. So ideally, validation should actually start with a documented method. Sometimes we can perhaps get into a cycle of development and evaluation, a sort of iterative process where we're trying to improve a method. But at some point, we need to take a decision and say, okay, I'm at the point now where I'm actually going to carry out a formal validation study, and really that should be against a documented method. This slide just shows a summary of what might go into a method description. So the title of the method that we're validating, a reference number for that method, if applicable. And then we should be clear on the status of the method. And I think Lawrence mentioned some of the categories of measurement procedure that are highlighted in ISO 17025. So we may have standardized methods that have been published, and effectively we're just using them without modification. But often there are modified methods that we use, and of course there may be methods that we've developed wholly in-house. And the reason it's important to understand this is that it will impact on what we actually include in our validation. And then it's also useful to document why we're carrying out this particular validation study. So is it a full validation, maybe of a brand new method that we've developed in-house, or perhaps it's a verification where we're using a method, a standard method without modification, and we just want to check that the method does work satisfactorily within our own laboratory. So just looking a bit more about the purpose of the study, there are a number of options. As I've mentioned, it could be a full validation of a method developed in-house, the verification of a published method where we already have some data on the performance characteristics or information on the performance characteristics. But sometimes things change. So maybe we've perhaps at some point carried out a full validation of our in-house method, but there's been a change in the scope of that method. So then we have to revalidate under that new scope. Something else might change around the method. So maybe some change in operating conditions, which requires us to reassess whether the method is still fit for purpose. Another situation might be perhaps if a method has not been in use for a period of time, then we would need to check that the method is still working correctly once it's brought back into use. So as I said, we need to understand these issues to help us plan an appropriate study. We also need to have an understanding of what our particular method is actually going to be used for. And this is often described as the analytical requirement. So this is effectively setting the scope and the parameters of operation for our method. And there are a number of things that we should really specify. So we need to understand what it is we're actually measuring. So what chemical or maybe biological entity it is that we're actually trying to analyze. So that might be specifying a particular element or elements, or perhaps a particular compound, or maybe a particular species or oxidation state for a particular element. But we also need to look in a bit more detail. So just specifying the analyte itself isn't really sufficient. So we can think in a kind of bigger picture about the measurand, and that's the quantity that we're intending to measure. So as well as knowing what it is we're actually analysing, we need to think about how much might be present, and are we interested in perhaps the total amount of that compound or analyte that's present, or maybe it's the amount that's extracted perhaps under specified conditions as defined by our measurement procedure. We also need to be clear on the measurement units that we're going to use for reporting. So here we're trying to put in as much detail as possible as to what our method is going to be used to test. We need to know what the samples will be. So what is their matrix and what physical form will they take? And also, why are we making the measurements? That can impact perhaps on decisions around the performance characteristics and what level of performance is actually required. So of course, there's a whole range of reasons why we might make measurements, but some typical examples might be to check compliance with a particular regulation. It could be part of maybe monitoring a production process. It could be checking compliance with a specification for a particular material, or perhaps it could be part of a research and development project. So when we specify the analytical requirement, we're really trying to give as much information as possible about what we're measuring, what it's going to be contained in, and what the results will actually be used for. I think when people think about method validation, I guess their main focus is often on the performance characteristics for a particular method. So when I ask people perhaps to describe what the validation process involves, their thoughts might immediately turn to the laboratory activities and trying to understand something about how their particular test method is actually performing. So there are a range of different performance characteristics that give us different information about different aspects of our method performance. So it's a fairly comprehensive list here, I think, but we can see that we don't necessarily need to study each of these parameters in each situation. Typical ones we might look at are perhaps around selectivity. So this relates to being sure that we're measuring what we think we're measuring and that we're not having issues perhaps with interferences. Often we're interested in the detection capabilities of our method. So looking at the limit of detection, so finding the kind of threshold where we can actually be reasonably confident that if the analyte was present, we would be able to detect it. But also in many cases, it's important to know at what level we can actually report reliable results. So that's often referred to as the limit of quantitation. We often need to know the range of application of our method. So perhaps over what concentration ranges the method will actually give results that are fit for purpose. And also perhaps something about the relationship between the response of our method and the amount of substance that might be there. So have we got a linear relationship, for example? Trueness is often very important. So this is really looking at our systematic errors and seeing how close our results are to the true value for the quantity that we're measuring. And also precision, which is looking at the random errors. It's looking at the variability in our measurement results. So there are many tables like this in the literature. They vary, I guess, depending on which sector they relate to. This one, I think, is adapted from a pharmaceutical standard for validation of pharmaceutical assays. So it's talking about looking for impurities and also main components, but you can get the idea depending on whether we're just interested in identifying something. There might be quite limited parameters that we'll study there, but in the presentation earlier today, Ricardo talked in quite a lot of detail about how we might look at the performance of effectively qualitative tests. And then these other columns relate to quantitative testing. If we're looking at something which we expect to be there at very low concentrations, then issues around capability of detection become important. Perhaps if it's a main component, then perhaps the quantitation limit's not so critical. So as I said, one of the reasons we need to be really clear about why we're making measurements and what that data will be used for is to help us to make sure that we choose the appropriate performance characteristics when we carry out our validation study. So once we've chosen our performance characteristics, we then need to come up with a detailed plan as to how we're actually going to evaluate them. And there are some common questions that we need to answer. So we need to think about what it is we're actually going to analyze. So what are going to be appropriate materials? And that will vary across the different performance characteristics. So for example, if we're trying to evaluate trueness or bias, we need a reference value. So it'd be quite common to analyze a certified reference material to help us to evaluate bias. If we're interested in precision, then we're looking at variability. So we don't necessarily need that external reference point. So potentially, if we're evaluating precision, we could perhaps use some surplus test materials. So we need to be clear for each performance characteristic as to what would actually be a suitable material for analysis. We then need to think about how many measurements we're going to make, and there is guidance available, often based around sort of statistical theory as to what would be a suitable number of measurements. So for example, to get a reasonable estimate of a standard deviation, which might be then used to express precision, then somewhere maybe between around 6 and 15 measurements might be considered suitable. Of course, there's always that element of balancing what's practical and sort of how reliable our estimates need to be. But we need to make sure we've got sufficient data to give us a reasonable estimate of the method performance. We also need to think about the order in which we're going to make the measurements. And in a moment, I'll just touch on some typical experimental designs that we might use as part of validation. And this is all part of the planning process. Obviously, it's very easy to go into a lab and just make lots of measurements, but that might not be the most efficient way of doing things. We need to be clear about the level of replication, the order, and also what data analysis we're going to apply at the end. There are various statistical tools that we can use, but unfortunately it'd be quite easy to generate data that then perhaps you can't evaluate in maybe the way you'd originally hoped. So it's good practice to actually think about what statistics you want to apply, and then make sure that the data that you're going to generate is actually suitable for those tests. So there's some specific tools that we might use in validation. So regression is obviously very common, particularly looking at issues around linearity. We might use analysis of variance, which I will illustrate in a moment. So particularly if we're looking at precision, but also we might use other significance tests as well to help us to establish whether we might have any bias in our measurement procedures. And then finally, we have to decide how we're going to assess fitness for purpose. So how do we decide whether our method is actually fit for purpose? What rules are we going to use? So I won't go into detail here, but choosing an experimental design is one of the key steps of a validation plan. We want to maximize the information obtained from an experiment. And if we plan things well, it may be possible to obtain information on more than one performance characteristic from the same set of data. So some common designs we might use are a simple replication, a nested design, fractional factorial designs, and also our linear regression or linear calibration. So simple replication is literally where you just make repeated measurements on a single material, and then we can calculate the standard deviation of those results. So this can be particularly useful for precision studies, and especially where we're perhaps just looking at repeatability, where measurements are made one after the other under very controlled conditions. If we happen to be analysing a reference material, then we could potentially also evaluate bias from that data. Slightly more complex design is our nested design, which I've illustrated here. So this is where each level of a particular factor appears only in a single level of any other factor. And where you might see this in validation is particularly where you're trying to evaluate precision over varying conditions. So maybe over different days or using different analysts. So here we have replicate measurements that are nested, for example, within an analytical run. And because we've got groups of data here, we can use the analysis of variance technique to actually look at the variation both within our groups and between our groups of data. So from this type of design, it would be possible to evaluate both repeatability, if these groups of data are made under repeatability conditions, but also we can look at the extra variation from one group to the next, and that would allow us to evaluate our intermediate precision. And then the other one I've flagged up there with fractional factorial designs, and that's where we have a carefully chosen subset of combinations of parameters and levels. And a common experimental design that's used in validation is around ruggedness testing. And what's actually shown here on the slide is a 7-factor Plackett-Berman design. So this is allowing us to look at 7 different experimental parameters at 2 different levels, and that's in 8 experiments. So that can allow us to perhaps screen to see whether there are some particular steps in a method that are critical. Performance targets, so this is often one of the more challenging areas. We need to establish some targets to allow us to be able to assess fitness for purpose. And there are a number of places where you might find such information. It might be defined in a particular standard or regulation. So I know certainly in the United Kingdom, around some of our environmental testing, laboratories are allowed to use particular, any test method really of their choice, as long as they can demonstrate that it meets particular targets, usually around precision and trueness, and also possibly around limit of detection. Sometimes it might be specified by the customer. So maybe your customer has actually had experience of similar measurements and has some idea about what level of performance is required. If you're using a standard published method, then often there will be information in there, particularly around precision. Sometimes you might be able to make a comparison perhaps against a similar measurement procedure. So if you've got other methods that you know are fit for purpose and have been used perhaps for similar types of measurements, then you could perhaps base your assessment on that. There is, I suppose, a special case where maybe if you are working in a very novel area and you really don't have anything else to compare yourself against, then you might just actually be trying to determine what's the current state of the art. So what is the method actually capable of? Finally, as I said, the validation process is not really complete until it's all been approved. So the validation plan itself should be approved before the work starts. And then after the study is complete, it's helpful to provide a summary of values and other information obtained for each of the performance characteristics, and also an assessment against the performance targets. And then there should be a final statement on whether the aims of the study have been achieved, and a declaration of whether the method is fit for purpose. And there should be a final sign off of that validation report, which would then effectively allow the method to be used routinely. So where can you find some further guidance? Well, you know from earlier presentations that we have our sort of main guide around fitness for purpose of methods and validation. And then we have this supplement, which is all around planning. So some of the issues I've talked about here, so around the template and how you plan your experiments is covered in more detail within that supplement. And as with all our documents, they're available from the Eurachem website. So just to sum up, so validation should be a planned and documented activity. For each of the performance characteristics we've chosen to study, we need to specify the materials we're going to analyse, the number of measurements, the order, data analysis, and the target performance criteria. I would recommend using some kind of template for a consistent approach, and that also allows easy conversion into a validation report. And both the plan and the final report should be signed off.